Alright, so this is going to be an overview of the BRCA1 gene presented by me, Hope Blaubin. So the BRCA gene is named that way because it stands for breast cancer 1 early onset. It is found in humans, although it is also found in other animals. However, for the purpose of this presentation, I'm only going to be referring to the presence of it in humans. It has 24 exons. 22 of them are coding and 22 are non-coding. There are some alleles of this gene, and the phenotype of some of these alleles is that it predisposes individuals to breast and ovarian cancer, as well as some other cancers, which I'll go over in a bit. It is located on chromosome 17, Q21, so that's its address. That means it's going to be on an autosomal chromosome, chromosome 17. It's going to be on the long arm of the chromosome, and 21 is kind of just a description of where you're going to find it on that Q arm. It's going to be about 81,000 base pairs, so it's a fairly large gene. There's 2 alleles of this, and you get 1 copy from each parent. There's many variations. However, it's the inheritance of this mutation of this gene where you will see the phenotype I will be talking about. And it is autosomal dominant. So the BRCA1 gene is going to encode for the BRCA1 protein, which is involved in DNA repair. When there's a DNA double-strand break, it's going to promote repair by regulating homologous recombination. It'll also regulate the non-homologous end-joining repair pathway and act as a tumor suppressor. Right here, I put a diagram as a reminder of homologous recombination. So it's just when 2 homologs, essentially, they're going to mix their sequences and exchange genetic material and create new fully functional combinations. BRCA1 is involved in the initial stages of homologous recombination, where a double-strand break is recognized. It's going to recruit other proteins to the site of the damage. In the non-homologous end-joining pathway, DNA is going to be repaired without the need for a homologous template. This makes it more error-prone and more prone to mutations. So, since BRCA1 promotes homologous recombination, taking the non-homologous end-joining pathway, this can lead to instability, errors, mutations, and possible cancer development. When we look at the alleles for the BRCA1 gene, there's a few that catch our eye. So again, BRCA1 is autosomal dominant, so it means just having 1 mutated copy, meaning you're heterozygous, is enough to significantly increase risk for breast and ovarian and other cancers. And this is because the remaining normal copy can eventually become lost, inactivated, or mutated itself, which will lead to total loss of function. So these well-known alleles, there's 2 in specific to be pathogenic that I want to focus on. The first one is 185 del AG. This is a deletion of 2 DNA bases. This is going to cause a frame shift mutation. This is going to change the reading frame of the gene, and it will usually result in a non-functional protein. So the BRCA1 gene will not function and create a functional protein. The second will be 5382 INT C. So this is an insertion mutation. which also causes a frame shift mutation, and it disrupts the BRCA1 protein's ability to repair DNA via homologous recombination, just like any mutation, any pathogenic mutation you would see in one of these BRCA1 alleles. BRCA mutations are going to be autosomal dominant, meaning that it's one of our numbered chromosomes and not on a sex chromosome. And one mutated copy is enough to increase cancer risk. So we see here in this diagram a father who is heterozygous, carrying one copy of the mutation, and one mother who is not a carrier. And all these children, they have about a 50-50 risk of inheriting the mutation. So even though it's not sex-linked, we will get into this later, we most often see these mutations causing a development of cancer in women. But it's important to note the BRCA gene is dominant, and we are talking about cancer susceptibility and your likelihood of getting cancer and predisposing you to cancer, not in causing cancer directly. So if you inherit the mutation, you are not absolutely going to get cancer. It's just putting you at risk. Pathogenic variants in the BRCA1 gene are strongly linked to increased cancer risk, especially breast and ovarian cancers. So carriers of a BRCA1 mutation, when we look at this, we see that the mutations are responsible for about 40% of all inherited breast cancer cases. And when you look at breast and ovarian cancers, BRCA1 accounts for over 80%. Although BRCA1 mutations are more often discussed in women, as we often see it a bit more often, men are also affected. So male carriers also have an increased risk of developing cancers, specifically prostate cancer and male breast cancer, even though they're less common. Because of this strong link, there's genetic screening available, and I'll get into that in the next slide. I did create a chart showing cancer type and the risk for malignancy if you are a carrier of a BRCA1 mutation. So this includes breast, ovarian, male breast, prostate, and even pancreatic cancer. Luckily, we have genetic testing. So genetic testing is a really powerful tool we can use to detect BRCA1 mutations. This process starts, you take a sample, it's usually going to be blood or saliva, and then you isolate the DNA from the cell. The gene is then sequenced, and any detected variants are compared to databases of known pathogenic mutations to determine whether the BRCA gene is harmful. Testing is especially recommended for individuals with a family history of breast or ovarian cancer, those who have had these cancers themselves, and individuals of Ashkenazi Jewish descent, where we see BRCA mutations quite often, just due to genetics. If someone tests positive, they can then begin preventative steps such as increased surveillance, lifestyle changes, or even preventative surgeries to reduce cancer risk. And here are the sources I used to complete this research. Thank you for listening.